Okay, now we have scientific developments that was observed in microbiology. So the major contribution was from, as I told, it was Pasteur, whose full name was Louis Pasteur. He made many scientific developments in microbiology. Now, moving to the next major contribution, which was from Robert Koch, he told how perfectly you can study the microbiology. Then another contribution on on the introduction of antiseptic surgery was from Lord Lister. Then the fourth contribution, which I have noted down here, is chemotherapy, which was from Paulellis. Now we'll study each scientist's contribution that was being made. So, firstly, we'll see what is the contribution from Louis Pasteur. He's one of the trained chemists from France. He studied fermentation, how the fermentation took. Uh, then he studied about the different types of fermentation that can be observed from different kinds of microorganisms. Then he was also known as the father of microbiology. These are the main points that I've noted down here. And nextly, we'll see the important contributions that were made by Louis Pasteur. So, the first contribution that is being mentioned here is the development of methods and techniques of bacteriology. So, he developed various methods and the techniques to study the bacteria. Then, secondly, he proved conclusively that all forms of life, even microbes, arose only from their like and not de novo. And third contribution was Pasteur faced many challenges from Pouchet, who was a proponent of spontaneous generation theory of microbe. Pouchet was one of the microbiologists who propagated and who was a strong believer in spontaneous generation theory. But Pasteur disproved Pouchet's spontaneous generation theory that by demonstrating the ubiquity of microorganisms in the air by his experimentation performed in the swan-necked flask. So, to conclude, Pasteur disproved the spontaneous generation theory by using the swan-necked flask for the experimentation, which show the growth of microorganism that is the growth of microorganism happened only through the air. And fourthly, he introduced the sterilization techniques that is nowadays being used as steam sterilizers, autoclaves, and hot air ovens. So these are the different sterilization techniques that is being used nowadays also, which was a major contribution from Louis Pasteur. And fifth point is that he studied on anthrax, chicken cholera, and hydrophobia also. He's made various studies on rabies also, but he couldn't isolate the microorganism from man after the dog had bitten the man. But he after he observed the saliva of a dog under the microorganism, he was able to find out that it was rabies, but he couldn't isolate it from the man from man. So that was one of his contribution. And next, so the other contribution which he made major contribution which he made was the live vaccine. Nowadays also we use live vaccine, which is being attenuated for the prophylactic use. So, how did he discover this live vaccine? Was an accidental observation of chicken cholera bacillus culture, which was left for many weeks, and this chicken cholera bacillus had lost its pathogenicity, that is, its strength to kill or the cause infection was lost, and it had retained such an ability to protect the birds against the subsequent infection by them, led to the concept of attenuation and development of live vaccine. So, Pasteur, what he did was he left one of the culture which contained chicken cholera bacilli on a table. After a few weeks, he observed that under the microscope, he observed that there was such an ability from the chicken cholera that it could protect birds from frequent infection by this chicken cholera. So he developed this concept of live vaccine, and he did this live vaccine concept under a accidental observation. Then there is another live vaccine which he gave, which was anthrax bacillus, which was incubated at high temperature, and proved that the inoculation of such bacilli in the animal induced specific protection against anthrax. So he bought that anthrax, incubated at high temperature, and inoculated the bacilli into the animals. So after he inoculated into the animals, he observed that this anthrax has developed a strength to protect the protect the organism from the anthrax bacillus. Then this is how he accidentally invented the live vaccine. So he had to prove it somehow to the public that this vaccine was a successful one. So he did one public demonstration in 1881, wherein he vaccinated the sheep, goats, and cows with the anthrax with the anthrax vaccine. So when these goats and cows which were given anthrax vaccine, they survived. They survived against all those unvaccinated animals. So this is how we prove that this live vaccine did had a prophylactic use. So moving on, Pasteur had major contribution of Pasteur was that he developed a vaccine for hydrophobia. So hydrophobia is who is scared to water, who doesn't like water, and who shows tremors and uh, what scaredness towards the water. So he had developed a vaccine for it also. It was a big milestone in the development of immunization in the medicine. Moving on, we have Joseph Lister. So Joseph Lister was a German general practitioner. He was a professor of surgery in Glasgow Royal Infirmary. Okay, he was a professor of surgery in Glasgow Royal Infirmary. He applied Pasteur's work in his experimentation and introduced the antiseptic techniques in surgery. This had given a very much effective pronounced drop in the mortality and morbidity due to surgical sepsis. So the introduction of antiseptic techniques had very drastically decreased the sepsis that had happened. So because of sepsis there were many people who were dying. So because using this antiseptic to clean the contaminated hands, it had reduced the death rate. He is known as the father of antiseptic surgery. Moving on, we have Robert Koch, a German general practitioner. Uh, he is also known as father of bacteriology. So he perfected his major contributions were he gave a perfect format of studying a bacteriological techniques and introduced various methods that can be used to isolate the bacteria from pure bacteria which were the pure strains of it. Then he introduced various methods of obtaining bacteria in a pure culture using solid medias. So we'll be studying what is solid media, pure culture, all other things in the subsequent videos which I'll be uploading. So just know that he had introduced a new method to isolate the bacteria or culture the bacteria from the solid media. And he also introduced the staining techniques, the different stains that were used to identify the bacteria from the surrounding field of microbiology, microscopic view. Then the fourth contribution, which we can observe, that he discovered the anthrax bacillus, tubercular bacillus, and cholera vibrios. He had discovered these new bacteria that were observed under microscope. So we have some Koch's postulate. So what is this Koch's postulate? It is according to Koch's postulate. A microorganism can be accepted as causative agent of an infectious disease only if the following conditions are fulfilled. So Koch had formulated certain conditions which determined whether the microorganism was a causative agent of infectious diseases or not. So the first condition was the organism should be constantly associated with the lesion of the disease. That is, the microbe or the organism should be should be present always in the lesion or the pus that is being formed. Lesion is a kind of pus or a discharge from that area of infection. Then it should be possible to isolate the organism in a pure culture from the lesion of the disease. So the pus or the lesion should be in such a way that you should be able to isolate the organism into a pure culture. Then the third point is the isolated organism, when inoculated in suitable laboratory animals, should produce a similar disease. Now, if I say if you take my organism from one cow which is being infected from anthrax bacilli, it has got anthrax. So you are taking the infected lesions specimen and you will be culturing it into the another organism, another cow, and you have to observe what are the different changes that is being observed in the other organism. When there is an infection or the lesion that is formed in the second animal, then that inoculation from that lesion should produce the same organism. Okay, so the fourth one is it should be possible to re-isolate the organism in pure culture from the lesion produced in the experimental animal. So I had told you that if you take a specimen from the second animal that you have put from the you have put it as an inoculation into that organism, you must be able to obtain the pure culture from the lesions of it. So these are the four conditions. Please do refer Koch postulate properly and write down the answer because it is an important question. They might ask you for 3 or 5 marks question. So do read the Koch postulate thoroughly. Moving on, we have Koch's phenomena. So, what is the Scots phenomena? How did it come? These postulates did prove that they were very important ways to determine the causative agents of infectious diseases, which we saw in the previous thing. Now, it wasn't. Though the postulates were satisfying, they did not. They were not able to be proved in all the cases. So, one such case was. In lepra bacillus. So Koch postulated a phenomena. Phenomena was being given. So what is this Koch phenomena? So this Robert Koch observed that the guinea pigs already infected with tubercle bacilli responded with an exaggerated inflammatory response when injected with tuberculous bacilli or its protein. This hypersensitive reaction is called as Koch phenomena. So this Koch phenomena is nothing but when a guinea pig was infected with tubercle bacilli, you again give bacilli to it. So when you give tubercle bacilli again to it, it gets more exaggerated, it gets more infected, and there's going to be more inflammatory response compared to the first infection or the first inflammatory reaction. So that was called as Koch's phenomena. Moving to the next scientist was Paul Ehrlich, who had made a contribution in chemotherapy. So Paul Ehrlich was again a German scientist. He was called as the father of chemotherapy, and we'll see what are the contribution that was made by Paul Ehrlich. So the major contributions were he applied the stains to cells and tissues for the study of their function. So he applied certain stains to the cells and the tissues to study their actual functions that was happening inside the cell. Then he also reported that the acid. He also reported the acid-fast nature of the tubercle bacillus. The acid fastness nature of a typical bacilli was observed, and he proposed a side chain theory of antibody production. We'll see what is side chain theory in the subsequent classes also. Just know the term side chain theory of antibody production. And then discovered salvarsan. Salvarsan was an arsenical compound which was used as a magic bullet. It was capable of destroying the spirochete of syphilis. So spirochete is one of the bacteria's important organ. Uh, Not an organ. It is used for movement. So spirochete was destroyed of the syphilis. Then he also discovered the neosalvarsan, which again gave a new branch of medicine known as chemotherapy. So neosalvarsan was the very first step to the discovery of chemotherapy. And the fifth point, which was the sixth point, was that he introduced methods of standardized toxins and antitoxins. So these are the major contributions that Paulellis made. So there are some important discoveries of other scientists also that are being given, which is lepra bacillus, which is Hansen who discovered the lepra bacillus. Then we have gonococcus, discovered by Neisser. Then we have diphtheria bacillus, discovered by Klebs and Loeffler. Then we have pneumococcus by Frankel, meningococcus by Wishelbaum. Then we have diphtheria toxin, Rox and Yersin, tetanus bacilli by Kaitsato. Then we have plague bacilli by Yersin. So these are the important discoveries by other scientists. Please do look into the major contributions of each and every scientist that I have been given as a points in the video, because they might ask 3 or 5 mark question. There is always a question on contributions of the scientists that I've told about, Paul Ehrlich or Koch's phenomena, which can be given for 3 marks, or the Koch's postulate, which can be given for 3 or 5 marks. You can observe that in various question papers, Koch's postulate has been the major question that has been asked. Then we have Joseph Lister. You must know what was his contribution also. Then Louis Pasteur's contribution. Yes, Louis Pasteur's contribution was also a major question that was being asked, that is being asked very frequently in the question papers. So this completes the video on history of microbiology. And in the next video, we'll be studying about microscopy and morphology of bacteria, which is also a very important chapter. So we'll see what are the different types of microscope that are available, and we'll study what are the different structures that are present in the bacteria. It will be more about bacteria itself. So I'll be dividing it into types of microscope and morphology of bacteria into separate videos. Please do subscribe to the channel for more update on the videos that I'll be uploading in the subsequent time. Hope you like the video. Bye bye.